Thank you for joining us and discussing how new technology advances with next-generation sequencing are enabling us to change our approach in tumor profiling and increase our knowledge about cancer. In oncology, we are, again, at a crossroad where single gene testing can no longer provide us with the information we need to fight the illness. Let's explore the reasons behind this change and the solutions now available to you. In traditional pathology, cancer used to be considered a disease of the organ or the tissue. This is still true, and it has not completely been replaced by molecular taxonomy, but we now see relevance in characterizing a tumor further. A lung cancer sample can now be further characterized as positive for a mutation in the EGFR gene called L858R, for instance. You can see here on this slide multiple examples of organ tumors where we now have a better understanding of which mutations are present and potentially driving the carcinogenesis. The molecular profile paints a much more comprehensive picture of the tumor and provides information on prognosis and treatment selection. Owing it in large part to early pulse-chase phosphorylation studies, as well as genomic studies associating genotype and clinical outcomes, cell signaling pathways and genetic drivers of cancer are pretty well characterized today. On this slide, we see the HER2 pathway, which is very important in breast cancer. 35 to 40% of patients expressing HER2 receptors will show a strong response to a drug that prevents the dimerization of cell surface HER2 and therefore shuts down the pathway activation and uncontrolled cell division. HER2 was one of the first pathways to be targeted by a drug, selectively on the HER2 expression level of a patient. So knowing about the tumor profile for HER2 in a specific breast cancer sample has enabled targeted therapy, selecting the right drug for the right patient. However, the cell has a myriad of complex circuitous pathways that allows it to quickly bypass a shutdown path through another means. What this means clinically is that you'll see a strong positive effect after the start of the first targeted therapy, and the patient will go into a dramatic remission. Unfortunately, in certain cancer types, the patient may develop resistance to this drug pretty quickly to another pathway activation, and then the patient relapses. So we will need to shut down the pathway at another point using another targeted therapy, hopefully leading to a second remission, typically less dramatic, and then see a relapse again. This may happen for a few cycles until the patient succumbs to disease. This is the current state of treating advanced cancers. To make the matter worse, the targeted therapeutic arsenal is rapidly growing, but only still a fraction of the current pharmaceutical options available. When those targeted drugs are benefiting the patients, the tumor cells can still become resistant. So this basically means today that we're doing all this drug development work for a subpopulation of cancer patients, and it will work only for a limited period of time. This is clearly an unsustainable strategy. Let's discuss targeted therapy as we know it today in a little bit more detail. It's called companion diagnostics when the drug and the tumor profiling test have shown clinical benefits together in front of regulatory bodies. The drug targets a specific cellular pathway, and the test is assessing the presence or absence of a mutation or the expression of a receptor, as we saw in the example with HER2. The current paradigm is to start with a drug, test the tumor for a single target companion diagnostic test, and then inform the patient whether he or she is a candidate for the companion drug. There are now over 25 drugs which include a companion diagnostic, and over 75% of the oncology drugs in development include a companion diagnostic or a complementary diagnostics and their commercialization strategy. With this number target, a lab may run out of tissue and budget, and the patient may run out of time before iterative testing can provide a comprehensive answer. And relapse is always an eventuality, in which case more information is then needed to find the next therapeutic option. So what solutions do we have today to gather as much knowledge as we can on the tumor in a rapid, cost-efficient way? By replacing iterative single-gene testing with a multi-target approach in one test, the lab can create a comprehensive molecular profiling of a tumor to help guide an oncologist's decision and the therapeutic management. By analyzing all relevant mutations present in a sample, drug response can be predicted and the treatment course can be optimized in a faster, more cost-effective fashion. So we discussed already how the use of molecular profiling can aid in treatment decisions. This brings value with the increasing number of targeted therapies available. Molecular profiling can also be a valuable diagnostic and prognostic information, as well as insight into the mechanism of resistance, and this is what we're going to discuss now. So we see here on the left an example with chronic myelogenous leukemia, CML, which is caused by BCR-ABL1 fusion gene. It can be monitored during treatment by quantifying allele burden of BCR-ABL1. When the allele burden rises and the patient relapses, it can be caused by a lack of compliance to medication or because of the rise of a resistance mutation, often the tyrosine kinase domain of ABL1, a mutation called T315I. Identifying the cause of the relapse, whether it's a molecular change in the tumor or a lack of compliance of the patient, helps the treating physician deciding the best course of action. Another example is here on the slide on the left with non-small cell lung cancer. On the right here on the slide. So it is known that resistance to first-generation tyrosine kinase inhibitors can be caused by the rise of a mutation in the EGFR gene, such as T790M or C797S. Since recently, there's a therapeutic options now available for patients that have relapsed because of the T790M mutation. Therefore, knowing not only the initial tumor profile, but also characterizing the tumor as a response to treatment changes brings incredible value in management of the disease. The current therapeutics and diagnostic landscape has changed drastically in the last few years and now requires comprehensive profiling in order to make treatment decisions. Single gene tests are just no longer sufficient. So what are the options for a multi-target approach? Illumina has recently released the TruSight Tumor 15 panel, and you can see here on the top the list of the 15 relevant genes to replace iterative tumor profiling with a single comprehensive test. The panel, as you can see, includes the usual suspects such as KRAS, NRAS, BRAF, EGFR, KIT, and a lot more. The gene content was informed by industry-published guidelines like NCCN and ESMO, along with input from key opinion leaders and our pharmaceutical partners as far as which targets are potentially relevant in the near future as well. TruSight Tumor 15 panel delivers comprehensive sample-to-data solution with an optimized workflow. The assay provides highly sensitive detection of relevant somatic mutation in solid tumor with a minimum of 5% variant allele frequency at over 500x coverage. This is compatible, very important, this is compatible with formalin-fixed paraffin-embedded tissue. It only requires 20 nanograms of DNA for the test. It has a quick turnaround time of less than 36 hours. So beyond the increase in targeted therapy drugs and the inability of single gene tests to provide a comprehensive tumor profile, there are several logistical reasons as to why single biomarker testing is just no longer an efficient avenue. The first reason is time. Turnaround time is of paramount importance in oncology testing because delays could result in less than optimal outcomes. For the lab, iterative testing can be very time-consuming. I show here an example for colorectal cancer sample. The one-gene testing used to be sufficient with KRAS. However, it's now recommended to test for at least 3 genes. So sequentially testing 3 genes will take up to 3 full days in the lab, while a single test can provide information on 15 genes, meaning for more than colorectal sample, but potentially for other solid tumors to be run at once, providing answer in less than 36 hours. So taking an iterative approach takes up precious time for the lab and precious time for the patient. A second logistical reason why a panel approach makes more sense than sequential single-gene testing lies within the sample. Retrieving DNA from a FFPE sample in acceptable conditions and quantity for molecular testing can be quite a challenge. The TruSight Tumor 15 panel only needs 20 nanograms of FFPE DNA to provide results on 15 relevant genes. By contrast, each single gene may require over 10 nanograms of DNA for a total input of material that can easily exceed 50 nanograms for gene testing. Preserving your precious sample with a next-generation sequencing panel means a little DNA input and a maximum output of useful information. So we discussed the logistical value of reducing turnaround time, batching solid tumor of different cancer types into the same run, and preserving precious biopsy material by using minimal amount of DNA in a single panel instead of multiple tests. Here's now another practical argument in favor of panel testing. There's a tremendous cost in sequential testing. Here's an example per tumor type of the cost of reagent and technical time required to perform sequential single gene testing. It's based on the recommended genes for colorectal, so you see here on the left, so including KRAS, NRAS, and BRAF. We have the example with melanoma with KIT and BRAF, and non-small cell lung cancer, which includes a number of genes, EGFR, KRAS, RET, MET, and BRAF, and those are the ones included in the TruSight Tumor 15, and leaving aside the FISH technology. So as you can see in the last bar, you can see that the same information can be obtained with next-generation sequencing panel versus doing iterative testing, and it shows the associated cost with it. Using a panel approach is an economic choice for multiple tumor types. So let's now have a look at which panels are available today at Illumina that can support your oncology needs. We'll look more specifically into targeted sequencing over the broader approach of whole exome, whole transcriptome, and even whole genome solutions that are also available at Illumina. Those targeted panels that you see here on this slide are for research use only. Let's start with germline mutation testing. There's a panel called TruSight Cancer. It detects 94 genes and an additional 284 SNPs to help identify inherited causal variants related to cancer onset. For somatic mutation on solid tumor, Illumina has a new TruSight Tumor 15 that we recently discussed. With targeted relevant 15 gene detection, this is the easiest sample to data workflow to transition from single gene testing to the comprehensive information provided by next-generation sequencing. For somatic mutation on blood cancer, the TruSight Myeloid panel can help identify variants in 54 genes that are related to hematological malignancies. More recently, Illumina also released the TruSight RNA Pan-Cancer, and I think this came out last month. It investigates variant callings, gene expression, and also gene fusion, regardless if the fusion part is known or novel. So in this presentation, we've discussed the changing oncology landscape and showed the need to replace single gene testing for a broader, more comprehensive understanding of the tumor molecular profiling. Using a targeted panel approach can help save precious time, biopsy material, and cost by increasing the lab's efficiency. Illumina has various targeted solutions available to accompany your lab in this transition from single gene testing over to a multi-target approach. I hope this information was helpful. We'll now open the presentation for questions before my colleague Claire goes into more details about the panel workflow and their use on the brand new MiniSeq.
Thank you, Maude, for your excellent presentation. We will now move to some Q&A. As a reminder, phone lines are listen only, but you may enter questions using the Q&A box at the bottom right of your screen at any time. And now we are ready for our first question. Maude, as guidelines change, how scalable is next-generation sequencing?
That's a great question. We've seen it in the last few years with different tests that have come to market only to see guidelines changing and our understanding of the relevance of certain genes and the clinical importance of knowing the status of other mutations, making those tests obsolete very quickly. I think next-generation sequencing is scalable to the intent that the panels, the current, for example, TruSight Tumor 15 now includes genes that are relevant today, but also targets that are in phase 2 and above from different pharmaceutical partners. So the idea is that some of those genes may not have a place in the guidelines today, but this is something that we see potentially could change in the future, and then the panel is already adapted to decide to include those or not. So potentially, next generation sequencing is of course evolving, and you will see panels becoming, we've seen a trend where panels are becoming larger and then becoming smaller again, just to be targeting the most relevant mutations. So we've definitely gone through a little bit of a trial before in next generation sequencing use, but it seems now that smaller relevant panels are definitely finding their place in the routine testing. So it is scalable to a certain degree, and I think having the flexibility to add targets that are relevant for pharmaceutical partners today is also increasing the flexibility of the panel.
Okay, great. Thank you. Another question that just came in. At what point is doing whole exome sequencing or RNA-seq more economical than NGS panels?
That's a good question, and it really depends on the information that you're looking for. So depending, of course, on the alternative technology, for example, with RNA sequencing, the different gene fusions, the economical aspect of it will really depend on the alternative method. So whether FISH is being used or any other technology is being used to detect those fusion genes. So I think right now we're getting to a point where next generation sequencing cost efficiency has been tremendously increasing in the last few years, and is definitely rapidly growing in use and effectiveness. So it'd be really hard to make a specific point as far as when it makes sense. But what we've seen in a lot of cases is when we're testing more than 2 to 3 genes per cancer type, it becomes very interesting to consider an alternative to single gene.
So now I would like to turn the time over to Claire Ray, Associate Product Manager of Oncology at Illumina.
Thank you, Kelly. Today I will kind of following Maude's presentation about what can benefit using NGS testing over single iterative testing. I'm now going to talk about what sort of products Illumina offers, and in particular how these products are being used on our new MiniSeq system. The products that I'm going to focus on today are revolving around our tumor profiling workflows. So I'll first talk about our TruSight Tumor 15 application. This is our solid tumor, very targeted library prep solution. I'll then talk about our TruSight Myeloid resequencing panel. This is targeting on hematology. I will then talk about our TruSight RNA Pan-Cancer panel. This is one of our new RNA pan-cancer targeted panels released last year. And then I'll finalize with our custom option, our TruSeq Custom Amplicon low input solution. Okay, so first talking about TruSight Tumor 15 and its MiniSeq Workflows solution. So TruSight Tumor 15 is a multiplex PCR library prep assay. Library prep time takes 7 hours to complete. If you start on day 1, you can finish up the library prep and then put it onto the MiniSeq sequencer to be completed with sequencing on day 2. Sequencing itself takes about 24 hours. It's a 2 by 150 cycle run. Analysis can then be kicked off using either a cloud option on BaseSpace using our TruSight Tumor 15 app, or you can use a local option on Local Run Manager, which is our new analysis solution on the MiniSeq platform. Overall, from sample to analysis, it takes either 2 to 3 days, depending on how quickly your lab wants to work through the protocol. So with TruSight Tumor 15, as I mentioned before, this is our solid tumor assay. So it's been designed and optimized to be compatible with FFPE DNA. The input requirement for TruSight Tumor 15 is 20 nanograms, and this is regardless of the quality of FFPE DNA you have. So whether it's high or low, we recommend a 20 nanogram input amount. It takes as little as 2 days to go from sample to data, with hands-on time mainly being on the library prep side, being an accumulation of 3.5 hours. Here you'll see listed the 15 genes that we're targeting using this particular assay. The library prep kit itself is a 24 sample kit. And with this kit, we recommend 8 samples per run on a MiniSeq. And so MiniSeq has 2 different run options. You have the high throughput option, which produces 25 million reads, and you have the mid throughput option, which is produces 8 million reads. And so when running this on a MiniSeq for TruSight Tumor 15 on the high output option, we recommend 8 samples per run, and then on the mid output option, it would be 3 samples per run. Sequencing time takes approximately 24 hours because of the 2 by 150 cycle run. So with the 15 genes that we target, it spans across 7 different cancers. And so these genes are targeting cancers such as breast, melanoma, gastric, lung, ovarian, colon, and prostate. Since this is a solid tumor assay, somatic variation is very important. So with this assay, we want to make sure that we can detect variants down to a 5% variant allele frequency. We also recommend a coverage of minimum of 500x, and that's across all assays. And so for this particular assay, the specification that we give for coverage is we want at least 97% of all content of all bases to be covered at a minimum of 500x. So as I mentioned before, TruSight Tumor 15 was released at the end of last year. Since then, we've had several customers provide data feedback for us. And here's some results from our beta testing that we had performed across 5 different sites globally. And so with these 5 different sites, we asked them not only to test control samples where we had known variants of significance, but we also wanted them to run any sort of FFP samples they had that had previously been tested using a different either single gene or NGS method. So what we had from 5 sites, we had 55 samples, and these 55 samples were previously characterized on one of the either PCR, real-time PCR, pyrosequencing, a different NGS panel, or Sanger sequencing. We then took these samples and ran them with the TruSight Tumor 15 assay, and then we compared the results. What we found was there was 100% variant concordance between TruSight Tumor 15 and a previously used method. The only 2 discordant cases that we found were actually using the single gene method. So here we had an example where a customer had run real-time PCR on a sample and they found a positive BRAF mutation. Upon testing it again using the same methodology, real-time PCR, they found that this was actually a false positive, which was concordant with the TruSight Tumor 15 results. At a different site, we had a customer that was running pyrosequencing, and they found a sample with a positive EGFR mutation. Upon running, they took that sample and they ran it through real-time PCR, and the results they found was a false positive, which was concordant with TruSight Tumor 15. Another unique thing about TruSight Tumor 15 that we haven't done with our assays before is to create this predefined report. So what this predefined report is doing is it's taking all the variants that were found in your sample and it's comparing it to a list of about 800 known variants of analytical significance. And if these variants match, they're included into this PDF report. And they're annotated, giving the amino acid change, nucleotide change, variant frequency, as well as the transcript ID. This report is generated automatically whether you're using our cloud-based solution for analysis or our local run manager on the MiniSeq. And what this report does is a really quick and easy way to quickly assess what variants of significance are in your particular sample. And then if you want to do further filtering, you can then use our software of Variant Studio. Next, I want to talk about our TruSight myeloid assay. So our TruSight myeloid assay is another amplicon assay. It's based off of our TruSeq amplicon chemistry. With this particular assay, library prep is done in about 7 hours, so you can finish it in 1 day. From there, you can load it onto the MiniSeq instrument. Sequencing takes 24 hours because it's a 2 by 150 cycle run. Analysis can then kick off either in BaseSpace or using local run manager using the TruSeq amplicon analysis application. Some specifications around our TruSight myeloid assay. So with this assay, you're inputting blood, and so the input requirement is 50 nanograms. The entire assay takes about 2 days to go from sample to data, with hands-on time accumulating to 3.5 hours. There's 54 genes that are being targeted in this targeted resequencing panel. The library prep kit itself comes in a 96 sample kit configuration. And with this assay, we recommend 8 samples per run on a MiniSeq high throughput sequencing kit. And then if you're using the mid sequencing kit, it would be 3 samples per run. Approximate sequencing time is 24 hours since it's a 2 by 150 cycle run. This particular assay targets 7 different myeloid neoplasms. So here they are listed now. Since this is a tumor profiling assay, we're looking for somatic variants down to 5% frequency. This assay also has a recommended minimum coverage of 500x in order to make accurate somatic variant calls. So here's a list of all 54 genes that are included in TruSight myeloid. As you can see, it's a mix of full coverage of targeted exonic regions as well as some more targeted specific exons that have more known significant mutations. Just some highlights. So for the MPN disease, this panel detects DEC2, MPL, CALR, and then for AML, multiple genes are relevant, including CEP, CBPA, FLT3, IDH1, as well as 2 MLL partners and many others. So there's 2 different options for analysis when running TruSight myeloid onto the MiniSeq system. You can use both the cloud as well as the local run manager for the TruSeq Amplicon app. You can then, with the TruSeq Amplicon analysis application, you'll be producing alignment files as well as variant call files, or VCF files. These VCF files can then be put into our Variant Studio application, and in Variant Studio you can further annotate these variant calls. And you can compare it to Cosmic databases as well as the UCSC browser. You then can create your own custom report, and you can print out this report and use it routinely into your laboratories. All right, next I will talk about one of our newer panels, our TruSight RNA Pan-Cancer panel. This panel was released at the end of last year, and it is a targeted RNA sequencing panel. And this panel comes out of the technology that we use to develop our TruSeq RNA Access panel, but this is a more focused approach. And because it's more focused, we're able to make it more scalable using our desktop sequencers such as the MiniSeq. So library prep takes a bit longer. What you're doing is you're starting with total RNA and you're creating a whole transcriptome library. From there, you're enriching that library and pulling out content for about 1,300 oncology-focused genes. So library prep in total takes about 2.5 days, but once the library is created, sequencing and analysis kick off automatically. You can load this onto the MiniSeq system. It takes about 13 hours to go from sequencing, and then analysis occurs in base space, and it takes about 1 hour. So here's some specifications for the TruSight RNA Pan-Cancer panel. The input requirement for this is 10 nanograms of total RNA if you have high-quality RNA. If you have FFPE RNA, we recommend inputting between 20 to 100 nanograms, and this is dependent on the quality of FFPE RNA that you have. It takes about 4 days to go from sample to data, and that's mainly for the library prep. Sequencing and analysis are quite quick. And we are targeting 1,385 known oncology genes. The kit itself has 48 samples per kit, and the recommended throughput for the MiniSeq is 8 samples per high throughput run and 3 samples per mid throughput run. Sequencing time takes approximately 13 hours. It's a 2 by 75 cycle run. So the great thing about this assay is that you can get lots of different information from it. So not only are you able to detect RNA variants, but you're also able to look at gene expressions and, more importantly, gene fusions. So what is a gene fusion and why are they so difficult to detect? A fusion gene is a hybrid gene formed from two previously separated genes. And so this can occur as a result of three different situations. In the first situation, you have a translocation that occurs between two different chromosomes, which causes two unique genes to be fused together. In a different situation, you have a large chromosomal deletion, which causes two genes that are on the same chromosome to now be neighboring each other. And in the third situation, you have a chromosomal inversion. So you have a large section of your chromosome swapping and inverting itself so that you have two genes that are detected together. So with gene fusions, we're able to look at RNA sequencing to be able to detect what sort of large rearrangements are occurring in the chromosomes. And so I wanted to show a visual representation of how the TruSight RNA Pan-Cancer assay does this. And so with the TruSight RNA Pan-Cancer, what you're doing is you are enriching made libraries you're enriching for targeted regions that you're interested in. And so here I have an example where these lines represent libraries. And so with this library prep, you randomly fragment your RNA transcriptome, and you add adapters to each end. So these black lines are adapters. You then have 80 base pair probes that are biotinylated, which is what these are showing, that have sequences that are targeting the specific 1,300 genes that you're interested in. So in this example, we have a known gene, and it's having an unknown fusion partner. And so we are targeting that known gene within the enrichment panel for TruSight RNA Pan-Cancer, but we don't know what this particular gene fusion is. So during the enrichment step, as long as a probe is attaching to a portion of the insert, we pull that library out and we continue on to sequence it. And so here is an example of what this sequencing looks like. And so with our paired end sequencing, as long as we pull down a fragment of library that contains that fusion site, we're able to detect not only the fusion site, but both genes that are included in the fusion phenomenon. So here we have an example of 3 different reads that contain both the genes as well as the fusion site. And so when this is being reported in BaseSpace, BaseSpace will not only tell you that a fusion has occurred in this particular sample, but it will tell you which 2 genes are present in the fusion. So TruSight RNA Pan-Cancer has also been optimized with FFPE RNA. So here we show graphs showing high correlation between replicates of not just good quality FFPE RNA, but also poor quality FFPE. And with this assay, we use a quality metric called the DV200 to assess the quality of your RNA. And what the DV200 value is looking at is, in your total RNA, how many of the fragments of your RNA are larger than 200 nucleotides. And this is a way to assess how successful your library preparation will be. And so what this table is showing is replication concordance and correlation compared to what the DV200 value is. And so this is a very sensitive assay and very robust, even with very degraded FFPE RNA. All right, next I'll move on to our final library prep, and this is our TruSight Custom Amplicon Low Input Assay. So this is one of our custom Amplicon resequencing assays. It is an upgrade to a current technology that we had, our TruSight Custom Amplicon technology, and we've now made workflow improvements as well as lowered the input to make it more focused for FFPE DNA. So library prep has been shortened down to 6 hours, so you can complete library prep in 1 day. From library prep, you can then load it onto the MiniSeq. Sequencing takes 24 hours. It could take longer depending on how long your amplicons are. From there, analysis will kick off. Analysis can take up to 2 hours depending on how big your panels are. And there are options in both BaseSpace as well as the local run manager. So here's some specifications for the new TruSight Custom Amplicon Low Input Assay. And so you can start down as low as 10 nanograms if you have high quality DNA. With FFPE DNA, we have a recommendation of inputting between 10 and 100 nanograms, and this is dependent on, again, on the quality of FFPE DNA that you have. With this assay, we also sell now a FFPE-QC assay, and what this basically is it's taking a qPCR test to assess what's the amplification ability of your particular DNA, and that will dictate how much DNA input we recommend. So for that assay, we have a range between 10 to 100 nanograms of input. Sample to data can take up to 2 days, and hands-on time has accumulation of 3.5 hours. So with this new assay, this new upgrade to the TruSeq Amplicon workflow, we've also improved several of the library prep steps to make it more efficient. The main improvement that we made is replacing the filter plate to a bead-based method of filtering and purifying your DNA sample. And what this does is it now makes the assay automation friendly, as well as it condenses the time of library prep. We now have a new kit configuration for this kit. We offer both a 16 sample, 16 reaction kit size, as well as the larger 96 sample is still available for larger throughput labs. So with TruSeq Amplicon Low Input, you can do analysis both on base as well as local run manager. And with both of these options, use the TruSeq Amplicon assay, similar to what we saw with TruSight Myeloid. And the output files of TruSeq Amplicon, they give you both alignment files as well as the variant call files, or the VCF files. You can then take these VCF files, put it into our Variant Studio assay. This Variant Studio exists both on base as well as we have a local version of it that you can use on your MiniSeq. With Variant Studio, you could do further filtering, further annotation, and you can compare it to several different databases, such as the Cosmic database or PubMed. So with all 4 of these analysis solutions, we discussed data analysis and what your options are. For all of them, except for TruSight RNA Pan-Cancer, we have both a local and cloud-based options. And the main thing to focus on with both of these options is now you have one software that encompasses every single step it takes to run this particular instrument. So from sample setup to run monitoring, as well as run analysis, you just have to go to one location to perform all of these steps. So these workflow solutions that I discussed, these are only the beginning of what you can really do with NGS technology in oncology. And Illumina is the provider of your choice, is a provider of choice for your lab's needs in oncology discovery and testing. So from small and actionable targeted panels for solid tumor and hematological malignancies, to fusion genes with unknown partners, and then to whole exome, whole transcriptome, and now even immuno-oncology as well as liquid biopsies, Illumina's platforms and chemistries can really scale to your lab's demands. So we just touched upon what you can use with our new MiniSeq software, but here's just some examples of what you can do not just with the MiniSeq, but also with our NextSeq solution and our HiSeq solution.